So if it comes to here, today maximum will complete the lesson on this first part of this, how do organisms reproduce. So in last class, I told you what is the artificial propagation. Correct, guys? Artificial propagation of, so artificial vegetative propagation. So we are having three methods. One is what? Cutting. Second one is what?
Layering.
Layering.
Grafting.
So we are having the advantage of this artificial vegetative propagation. What is that? We can make more faster the fruits compared to the seeds. If you start planting the seeds and if you want to get the desired flowers or fruits, it will take a long period of time. But if you go with a vegetative propagation, what will happen? We can get very less attention in their early years than the plants grown from the seeds. And as well as we can get a seedless plants from the artificial propagation. And as well as single plant, we can grow what? Many varieties. And also we can, new plants we can replicate, means no need of any kind of seeds or anything to cultivate the new plants. So there are parent exactly like desired characteristics from the parent plant we can cultivate, we can grow in the new plant. Is it yes, guys? So like this we can produce artificial vegetative propagation exactly like the parent plants. Whatever the desired features of the parent plant will be replicated in the new plant. Means replicated means in the sense of it will copy exactly from the parent the desired characteristics, whatever we want. And also some seasonal fruits will be there. In that season only basically we used to get. But if we'll go with this grafting method, then what will happen? We used to get the fruits much earlier, only after few growing seasons itself. It no need to grow for a long period of time to get seeds. For example, like if you are planting a mango seed, it will take many years to grow and get like a matured stage and to form a new mangoes. But if we'll do with this vegetative propagation, even when the mango tree is very small, as yesterday I have shown you, is it yes, guys? Very small, it will start producing the mangoes. Yes or no? Yes. So now, nobody will answer. So now, till now, we all people are seeing that we have been doing the agriculture with the help of a soil only. So now, if you'll go for soil-less agriculture, then that is what the last technique which we are going to study here. That is what we used to say, tissue culture. So this is the most in future nanotechnology also will use here. So future, it is the most important thing in case of agriculture. Now also it is going on, but in the time of yours, these tissue culture will rule in agriculture. So major importance of this one. So let us comes to understand this tissue culture means. So nothing, as you people are studied, even class 6th, 7th, 8th, and even 9th, cell structure, plant and animal you people studied. Is it yes, guys? Plant cell and animal cell, respond please. So now if you notice it, so as we know, what do you mean by tissue? So the number of cells will together, they will form. Is it yes, guys? Together they will form organisms. Organisms, if they want to form, middle they will form what, guys? They will form a tissue. These tissues combine together and they will form what? Organs. And these organs only they will form what? Organism. So a tissues means in the sense of what we can say, it is a group of cells. Is it yes or no? So a group of cells combined together, that is what we used to call as tissue. Now, what do you mean by tissue culture means? This is also related to only for plants. What is this? Only for plants. Understanding? So now, what do you mean by this tissue culture means? We will get the new plants. We people can get the new plants from a small piece of plant tissue. That's all. So we'll get a new plants from where, guys? From a small piece of, that piece is only called as what, Arvind? Small piece of plant tissue. That is what we can say. What do you mean by tissue? The tissue is only called as cells. Do you understand?
Cell.
So a group of
Yes.
cells are from a small piece of plant tissue. We can we can produce a new plant from that small piece. That is what we used to call it as tissue culture. And here, no need to require any kind of soil at all. Even lab itself, we can cultivate millions of plants, new plants from the well-growing tips of the plants in a suitable growth medium. Understand? That is only called as tissue culture. So here, what is then without medium, like what we can say, without soil, how we can cultivate means. So we people will prepare a solution called as culture solution. So what is this culture? Tissue culture. Do you understand, everyone? So now we will prepare a certain kind of conditions which is required for amount of oxygen, amount of water, amount of like nitrogen, phosphorus, whatever it is required for the growing of plant. Everything will be in a proper ratio. That is what we can say, so a suitable growth medium. What we used to call the required conditions. All the conditions we used to make artificially. So suitable growth medium we people will prepare. That is what we used to call as culture solution. So no soil is required. Suitable growth medium we can say. So now here what will happen means, here we no need that cells also. What is that cells means? Remember, you have to take a growing tips is not required any kind of branches or stem or nothing. So tip, you people know that a very sharp edge, correct? Just freshly growing, new leaves are arising. Have you noticed it? Tips of a plant, please respond. So if this is the plant, if you'll consider for example, then tips will be where? Anybody can say, if this is the plant, for example, then where is the tips will be? A sharp edges, yes or no? So if we'll consider this is the plant, then where the tips will be? So this is what a sharp, is it yes or no? This is what a new leaves are arises. That is what we used to say shoot. Yes, guys? Please respond. So this shoot only we can take. That is what we can say, growing tips of a plant. Growing tips of a plants we can say. So these part we people will cut and we will keep it in a suitable growth medium. Then we people used to get new plant from that, and it is very fast. It will be even a lot of advantages of this technology. That is all we will study now. So here what will happen means, we people will prepare a certain kind of solution called as culture solution. It is containing all the required conditions for suitable growth. That is what we can say. All the required conditions for proper growth of plant. Do you understand, Karthikeyan, Dilepan, what I am saying? So now, here what we will do means, we will remove this growing shoot or tip of the plant, and we will keep it in this culture solution. That is what, where it is having a suitable growth medium. So automatically the growth medium used for growing plant tissues is very important in this process, because it is only contains what? All the plant nutrients, whatever it is required. The plant, like earth, soil, whatever it is providing, like nitrogen, phosphorus, potassium, NPK we can say. So these are the required main things, correct? So like all the required minerals, everything, all the plant nutrients will be, we people will prepare artificially. So that is why this culture solution is more important. So now these culture solution, it will be looks like in the form of jelly. You understand? It will be in the form of jelly. So this jelly is only we used to called as basically agar we can say. Do you understand? This culture solution only, it will be similar to a form of jelly. So that jelly is only the Hindi name we used to call as agar. And you remember, and also we will add, suppose you can see, you might be having a farmer, somebody, some your grandparents, or you may be having a agriculture land and all. So for well growth of plant, they used to add middle some pesticides, chemicals and all. Is it yes, guys? Please respond. Yes? Nobody will
Yes,
answer.
sir.
Yeah. So that is only we used to call as plant hormones. For example, see, somebody wants to get a very big size fruits. Somebody wants to grow the very fast growth of plant. So that is what, like ours also. Once we'll get mature stage, our new hormones will produce in our body. Is it yes or no? Like that. Plants also having certain kind of, they require certain kind of hormones. So the plant hormones which are necessary for the plant growth, everything we people will keep arranged artificially in this culture solution. All the plant nutrients and as well as required plant hormones. Required what is? Plant hormones.
Hormone.
Everything we people will make artificially a single kind of place where plant can get all the required things. That is what we used to call as culture solution. So now how it will happen, sir? So once you prepare a culture solution with the required suitable conditions of a plant growth, now we people will proceed and will make this tissue culture in a 5 steps. So let us see what is the steps and how we people will proceed. Shall we start? So first, what do you mean by tissue culture, guys? From a small piece of understanding, from a small piece of plant tissue, which is removed from the growing tip of a plant, we can make a new plant, we can production a new plants from that at a suitable solution. That is what a suitable growth medium. That is what we used to call as tissue culture. So now how this tissue culture will happen, that everything I will explain. So now you can see this is what we can say even soilless culture. So now observe, all of you observe these 5 pictures, then you people can understand what is 4 pictures. You can understand this tissue culture very clearly. So now what we people need to do means understand very carefully. So here, if you notice it here, with the help of microscope, what he is doing, guys, can anybody say? With the help of microscope, he is doing something. What is that? Anybody can say here? So what they're saying, a small piece of plant tissue they're taking from where? A growing tip. Have you noticed it, guys? This is what the plant, a growing tip. They have been cut everything. And you can see here, a small piece of plant tissue they are taking from the growing point of a plant. That is what the tip of a plant we can say. Have you noticed it? With the help of scissors and all, he is taking. Yes or no? Please respond.
Yes, sir.
Yeah, that is
Yes,
what.
sir.
So from the tip of the plant, he has been taking a small tissue from that. Now, they have been placed on a completely suitable medium. What is that medium called as? Culture solution. Have you seen? It is similar to like understand. So it has been placed in a laboratory where we can see a suitable solution which is contains all the plant nutrients and as well as plant required hormones. That is what we used to call as a jelly-like substance called as culture solution. Yes. Okay. So now you remember here this jelly is completely sterile medium. What do you mean by sterile in the sense of means it should be completely packed. It should not be affected by outside and inside gases exchanging and all because they are making in a particular, like you may be heard, surrogacy in humans. We are producing the babies in laboratory. Have you heard, guys? So the mother womb is not required to growth of a new baby. So like the same way here, it should be sterile, means in the sense of which contains all the requirements of plant, like plant nutrients and plant hormones, and there is no exchanging of gases and etc etc. Because if it is open surface, if it is not sterile, then what will happen? It may be affected by the some diseases. Is it yes, guys? Some bacteria, virus are maybe affected with that. So that is why it should be a completely sterile place, like isolate, fully it will be isolated. That is what you have to remember. So whatever the hormones and whatever the nutrients it is we have been given in these, so the hormones make the cells in the plant tissue divide rapidly and they will start producing many cells which will form a shapeless lump of mass. That is only you can notice it here. the fast because of whatever the hormones we are keeping in this jelly. So it will make this cells which are present in this, understanding a small tissue, it will make into a rapid cell division. So we used to get some shapeless kind of a group of cells. Have you noticed it here? Different kind of shapes we are getting. This different kind of shape which has been done, a shapeless lump of a mass, that is what we used to call as callus. What we used to call, guys? Callus, Callus, callus. So how this callus will form? Once you will keep this a sharp group of cells which has been taken, a small piece of plant tissue in this jelly. So we are keeping what it is, hormones we are keeping, yes or no, required plant hormones. So these hormones will influence this plant tissue to make a rapid cell division, and they will form a shapeless group of cells. That cells only we used to call as callus. Do you understand, everybody? Yes. So now,
Yes, sir.
these callus, what will happen means, you can see here, slowly it will be rearrangement. This callus, they will transfer to another jelly containing suitable plant hormones which stimulate the callus to develop into a roots. That is what. So once the callus will form, we people need to take this callus from this jelly, and we have to take another kind of jelly which is another kind of hormones which will stimulate. What do you mean by stimulate, guys? Which will encourage, do you understand, which will help to form a roots. So this callus, you can see, if you place in a new kind of jelly, suitable medium, it is used to produce the roots. Have you noticed it? This is what we used to say. So these all are called as roots only. Have you seen, yes or no? Please respond. That is. So first medium, it will make it into cell division, rapid cell division, and it will form callus. Now, second medium, if you keep, what will happen? These hormones which are present in this, they will help them to modify this replacement of the cells in the callus, and they will make them to get what, guys? The callus will develop what? It will develop roots. That is what it will happen. So now, once the roots will start developing, then this callus with the developed roots is put in another kind of jelly. Do you understand, Arvind? Nitinath? So we are keeping it?
Yes, sir.
Another kind of jelly. Jesse, are you understanding, Jesse? So now we will place in another kind of jelly. So still it contains a different kind of hormones where it will stimulate to developing shoots. Do you understand, everybody? So now, first jelly, what will happen? First jelly, what will happen? It will stimulate for what? Rapid cell division. What is this? Rapid cell division. So now, after cell division, if you place in a new jelly, the hormones which are present inside here, they will stimulate what? Formation of roots. Understanding? Now, after formation of roots, root callus, we can say rooted, the callus which is having roots. So now we are keeping in another kind of jelly where the hormones present in this jelly will stimulate to grow what, guys? Will stimulate to development of shoots. Do you understand, everyone? So the development of shoots will take place. That is what we can say. So now the callus having roots and as well as shoots. You can see here, it is having root and as well as shoot, both developed. So now we can separating into tiny plantlets. Do you understand? So they will start separating into individual plantlets. Plantlets means, you people know, in bryophyllum I told you. Do you remember? Plantlets means what? A small plants which will grow. Do you understand, everyone? So that bryophyllum plant, you can, do you remember, correct? So that small things which will come at the corners.
Yes, sir.
That is what we used to call as plantlets. So here also, same way, once the roots and as well as shoots will develop, then they will start forming into a small, tiny plantlets. Tiny plantlets. So here, many tiny plantlets are produced from just a few original plant cells. That is why this tissue culture is well popular structure. well popular. Now again, sir, after cell division, formation of roots, can you please explain? I saw okay, Kevin. Now you can see Kevin. So here, first of all, we will take certain plant cells from the growing tip of a plant. Growing tip of a plant. So we will take with under the microscope, we'll take very few cells, very sharp tips of the normal plant we can take. Now we will keep it in a certain kind of solution medium where it contains a certain hormones which will stimulate the rapid cell division, so that a huge number of cell divisions will take place, so that it will produce a hump. We can say that it is a lump of cells, like small mountains of cells. That is what we used to call as callus. So this callus is a shapeless body. So now once if you'll observe that callus is formed, so now you have to take this callus and you have to keep in a certain another medium where it contains another kind of hormones which will stimulate the roots. Do you understand, Kevin? So now callus will develop the roots if you place it in a new medium. Now, once if you feel the roots are developed in this, again we have to separate it from this jelly and we have to place it in another kind of medium where it contains hormones which will stimulate to develop the shoot. That is what it is. So once the shoot and root will develop, then the cells will start forming into a new plantlets. That is what we used to say. So a very few plant tissue, we can produce many number of small plantlets. That is why this tissue culture is a well popular culture. And here, no required soil for growing of new plants. That is why it is a more famous technology in the present centuries. So now you can see here, these plantlets, they will be produced or they will be transplanted into pots and soil where they can grow to form the mature plant. So once it will forming the shoot and root, once they are forming the tiny plantlets, then we people can separate it into individual parts. Each tiny plantlet, individual part it will keep, it will start growing into a new plant. Like that, this tissue culture will take place in 5 steps. Finally, we will separate it into a new parts or soil where it will cultivate into a new plants, individual plants. That is what we used to call as tissue culture. So now this tissue culture technique is the uses of this tissue culture technique is increasingly mainly for the production of a certain kind of plants called as ornamental plants. You people maybe know that decoration, stage decoration and all, they will do it during marriage time or during politicians are visiting. Have you noticed it? Some kind of orchids flowers and this chrysanthemum flowers and all, they will keep. Have you noticed it? Please respond. Yes?
Yes, sir.
So this tissue culture majorly we will use for production of ornamental plants because different varieties and fast growth we can make it. So that is why this tissue culture will use the production of ornamental plants like orchid flowers. You people maybe noticed it. Orchid flowers, you people know orchid flowers, and as well as dahlia flowers we can take, and carnation flowers, and as well as this all types of basically Bangalore you can see so many, even here also we people import the flowers for decoration, ornamental flowers from Bangalore only, because Bangalore and its surroundings were very famous for production of these kind of ornamental flowers, and a very cheap cost also there. But coming to here, it will be very cost effective. So this tissue culture method only, they will produce very fast growth of new plantlets, so that very fastly they can get the results, like in the sense of flowers. That is why they will follow this technique, because if they will plant the seeds, it will take very huge time to get the result. That is why they will follow these, and so that we can get very fast growth of plants and we can get extremely good results also. So now here, remember, here you can observe this tissue culture also we can call it the production of these plants. This based upon this tissue culture, we are taking the tissue, correct, a small group of cells we are taking. So that is why we can it is also we can call it as micro propagation. So the tissue culture also we can call it as what, guys? Micro propagation. Do you understand? So because of we are taking very small number of cells from the tip of the plant, that is what shoot, and from that we are developing the new plants. That is why it is called as micro propagation. Usually, even me and my wife also, when we are in college, when we are in our university, we also worked around 3 months on this tissue culture. We have been cultivated potato. Potato, you might be know that. So potato plants, basically we people will cultivate from the potatoes only. Yes or no? That we people will study. We have studied, correct. So potatoes, which will come under what? What technology are potatoes? You have studied, correct, last time. Bryophyllum, potatoes and all. What is the technology called as? Vegetative propagation. Do you remember? Nobody will answer. So these potatoes, new plants, basically we will get from the by cutting buds of the birds. That is what. But with the help of tissue culture, if you take the shoot of a potato plant, from that also we can develop a new potato plants. That is what we can say tissue culture, and that is what we have been done around 3 months in a biochemistry lab in our university. So that is what I'm saying. Very, very good, and it's a good feeling when we are cultivating like soilless, small, new kind of plants. And why, sir, you are following this method? What is the importance means? Remember, we are here. It is growing in a what kind of conditions? Arvind, Nitinath, can you say what kind of conditions it is? Completely sterile conditions. Yes or no? So there is no kind of diseases affected from the outside because no fungus, no virus, no bacteria will affect the plants. So what will happen here? We can cultivate, understanding, a disease-free plants. So if you follow the tissue culture, that is the most important thing you have to remember. So the plants which are coming here, they are completely disease-free. Do you understand, everybody? What I'm saying? They are disease-free plants. That is what you have to remember. And you can see, and very, very fast technique it is. Very, very fast technique. As I said, from the small group of cells, we can produce many kind of plantlets. So that is why thousands of plantlets can be produced within a few weeks time for a small amount of plant tissue. Very fast technique. These are the advantages of this one. So even you might be few of you are interested in agriculture, future you maybe understand what is that. Now, and another thing, this is it, we are keeping in a laboratory and a particular sterile condition. So everything artificially we people are providing. So is it can grow all around the year or only particular seasons, guys? Please respond. You people need to say, is it will grow all around the world, all around the year, or only particular season? Nitinath, Madhumita. So it is growing in a sterile conditions in lab. So we are not supposed to depend on weather or season. Whatever the season, whatever the weather, we can continuously throughout the year. It can grow all around the year. It will produce the plants. That is the next one. The plants can grow, understanding. So complete year, throughout the year, they can produce. That is what it is the advantage of that, not just the seasonal based. Next, you can see a very little space is needed. So if you want to cultivate a new kinds of plantlets, so we no need a very little space is enough to cultivate thousands of plants. So that is also matters now here. Even if you are having a small terrace, you people can start producing a new plant, supplying of new plantlets, like different kind of plants by a less space itself. These are the advantages of what it is a tissue culture. Understand? So that is why the tissue culture is a well popular technique in present days to produce the new plantlets. So this is what it is. Have you understand, everybody? Please, Kavya, Bhumika. So now,
Yes, sir.
what you
Yes,
will
sir.
do is please write the definition of tissue culture and later we will proceed. The last concept of this one, still it is there. That is what the beginning of your NCERT book, how do organisms reproduce, down the first paragraph and second paragraph and third paragraph, if you will see. Before starting of asexual reproduction methods, we're having something called as DNA transformation, and etc. in asexual reproduction. So why we need a sexual reproductive methods and what will happen in asexual reproductive methods, that is all the last one. That is the most important concept which the questions will raise us so many slides. That is what we will study now. So now tissue culture. So please all of you read the definition. Tissue culture. The production of new plants from a small piece of plant, plant tissue removed from the growing tips of a plant. In a suitable growth medium. Is called tissue culture. So what is this suitable growth medium used to called, guys? This suitable growth medium is only called as what?
Culture solution.
Culture solution. Culture solution. That is what you have to remember. Clear, everybody? Yeah. Please write it fast. This is what, please call the absentees here and let them to join. So now, if you'll understand this concept, now, as you people are already studied in lower classes and all, DNA, have you heard the word called as DNA? Deoxyribonucleic acid. Class 8th biology you might be studied. Class 9th also you might be studied. Cell divisions and all. DNA, deoxyribonucleic acid. And you might be watching movies also. If something they want to identify the father, they will go with the DNA test. Have you heard? That is what we can say genes. Genes. Have you heard? Please respond. So you might be have studied, yeah, the animal cell and plant cell. Have you heard? Have you studied animal cell and plant cell? Please respond. Yes.
Yes, sir.
Now, every plant cell and animal cell having a nucleus. So inside the nucleus, we are having a certain number of chromosomes. Have you heard of chromosomes? A word? Please respond. So the chromosomes only, the transformation of these chromosomes only from the parent to the baby. That is what a characteristics. If the chromosomes are transforming from humans to new babies, they will transfer the human characteristics. Plants. Like that. That is what we used to call as genes. Understand? So the genes, like we can say, like for even you can see at the time of our birth. You can say, this boy, you are like similar to your grandfather, similar to your father, similar to your mother, or else their grandmother. Like that, people used to talk. Have you noticed it? Yes. That is what we used to say, DNA transformation. So you all people, like particular family members, they're having a similar DNA structures. So it's like a carrying from one generation to another generation. That is what a transformation of chromosomes we can say. So now, if it comes to here, as we have studied till now, what it is, asexual method or sexual reproductive methods, asexual reproduction method, yes or no? So now, our doubt is here, is it the organisms which are producing in asexual reproduction, is it they are the exact copies of themselves? That is what our doubt. This is the organisms, they're creating the exact copies of themselves in asexual reproduction. Means in the sense of, like whatever the characteristics are there in like a parent, if the child is exactly, they are the copies of them. That is what, if the exact copy, what is the word we can use, guys? Replicate, yes or no? So they are exact copying. See, I can say he is the replicate of me, means in the sense of what? Replication of me. Similar, he is like me, all the characteristics of me, whatever I'm having, I am seeing in him. Then we can say he is just a replicate for me. Is it yes, guys? General language also we can say. So same way here, our doubt is now, if the things like maybe animals or plants, if they are following, if they are producing the new babies in the form of asexual reproduction, is it their exact copies of themselves? That is what our doubt. So now let us see the true it is not. That is all everything. So now the last concept we are going to study is, please keep heading, do organisms create exact copies of themselves in asexual reproduction? Asexual reproduction. So now little patiently you have to listen this concept very carefully. So this is more important and little difficult also to understand. So now if you want to understand this one, first you should know the meaning of DNA. So what do you mean by DNA, guys? Deoxyribonucleic acid.
Nucleic acid.
Understand? So what is this means, which will actually the genetic transformation which can identify the parent organism. So means a baby is coming from whom we can identify by transforming a certain kind of set of chromosomes from parents to the baby. That is what we can say DNA. That is what you people general language can say genes. So now here if you take here asexual reproduction, basically they will make genetically the production of genetically identical offsprings only. They will produce a similar kind of offsprings, but the genetical variations arises sometimes, understanding, occasionally inaccurate DNA replication. So during the cell division, if DNA replication is not done properly, if DNA copying at the time of cell division, due to some maybe conditions or due to maybe some new what we can say the situation